Okay, folks, let's talk about chapter number 3. And with this chapter, it's a really nice crossover to some techniques that we are going to do in the lab. And then also, this information, of course, comes from your textbook. So being tested on this content, both lab practical-wise, doing the labs and so on, and then also your first exam. So it's kind of a crossover between both lecture and lab. So we'll see a few tools, we'll talk about media a little bit, a few vocab words. So starting off, we have 5 basic techniques that we are going to utilize while we are in the microbiology laboratory. And we're going to start off with specimen collection. With specimen collection, on Tuesday, that's really the only day where you're going to obtain a specimen from an unknown, some random source, like your shoe or your watch or your phone. Every other class period, your specimen collection is going to come from whatever bacteria, we provide to you. Sometimes you'll receive a bacteria where you don't know what it is, and the purpose of your lab section is going to be to figure out what it is by running a series of tests. So that's pretty cool. We'll do some of that at the end of the semester. But starting off, we're going to learn all these techniques that you would utilize, and you'll have known bacteria. Now, the first term up here definitely is the most difficult out of these 5 I's. You're going to notice all 5 of these terms start with the letter I. The word inoculate. That means to take bacteria or a microorganism from one source and aseptically transfer to another source. So that would mean, in this little scenario, let's say, and this is just sterile media, so I'm holding it without gloves. So here we have, let's say for example, in this tube, this is E. coli bacteria. On this plate, there is nothing. So if I were to inoculate this plate with E. coli, I'll take a sample of E. coli from the tube, and I will aseptically place it on this plate. I just performed an inoculation. There's a number of steps you would need to do it aseptically. You'll do it the proper way, but that's what an inoculation is. So you can take bacteria from one source and transfer it to another. Up here, I also have, it is used to culture. The word culture is interesting because you can use it as a verb or you can use it as a noun. Culturing bacteria means you're making this bacteria, you're growing this bacteria. So I'll culture some E. coli by taking it from this source and putting it here. So I performed an inoculation, but I also did some culturing. So they're sort of interchangeable, so you can use culture as a verb. Using culture as a noun, this right here, remember there's E. coli in here, not for real, but we'll say there's E. coli in here. This is an E. coli culture. What I'm creating by doing the culturing is, in a few days, what I'll have here is a new culture of E. coli. Okay, so it would be a noun, because this would be an E. coli culture. But what I'm doing by making a new culture is culturing. Hopefully that makes sense. Okay, so you can use it as a noun or as a verb. So that is what we refer to as inoculation. And then this is a really nice diagram from your textbook. So specimen collection, again, other than the lab we just did on Tuesday, specimen collection will be coming to the front of the room, grabbing your plate or your tube of bacteria, bringing it back to your workstation, and doing whatever the lab has you do with it. Here we can see inoculation. So with inoculation, you're going to place your sample, your bacteria, transfer to another source, whether it's a plate, a broth, a slant, whatever it might be. We're not doing blood bottles or bird embryos. It'll always be some type of microbiological media. Okay, just like the sort of sterile ones I have up here, or the plate. The next 5I term is going to be incubation. Incubation is where we actually grow the bacteria. This is where we grow the microorganisms. So we have to allow this newly made culture to incubate. After you perform that inoculation, you need to have that bacteria incubate. So what we do is, for our incubation, you have to use proper temperature, and we like to use 37 degrees Celsius. What was the important thing about 37 again?
Body temperature.
Human body temperature. Oftentimes we use 25 degrees Celsius. Does anyone know what 25 is? Yep. Room temperature. Okay, so room temperature, human body temperature. Most bacteria like to grow between 20 and 40 degrees Celsius. Okay, that's the most common range for bacteria. Think about E. coli. Can E. coli grow on your countertop at room temperature? Okay, like food source, like you have, some type of protein like chicken. Yeah, of course. Can it grow within your body? Absolutely. Okay, so yeah, definitely, it'll grow. It's pretty hardy. Okay, it'll grow anywhere, in this range of 20 to 40. And of course, we have the incubators, and everyone placed their plates and their tubes in the incubator on Tuesday. Moving on to the other I's that we have: isolation, inspection, and identification. Oops, I went the wrong way. There we go. Okay, so isolation. Let's say you have this plate, and if you look at this plate, how many different types of bacteria do you see on that plate? Yeah, maybe 3. There's a few different colors going on up there. Okay. Bottom line, there's more than one. That's the bottom line. This culture, you would call it a mixed culture. So you should jot down your notes that a mixed culture contains more than one type of bacteria. Mixed culture contains more than one type. So let's say you have a mixed culture in front of you, and you didn't want all these bacteria. Let's say you only wanted the red one. Like, I just want to grow just that red bacteria. So you can do some isolation, and right here, we can see you can use this tool, and I'll pass this around in a few moments. But this tool, this is called our inoculating loop. So we're going to use it every time we do an inoculation. You hold it like a pen, you grab some bacteria from one source, transfer to another source. So we don't pour the bacteria or do anything else crazy. We use this tool for all transferring. So what you can do is be like, oh, I'm just going to grab the red one, and I'll just put the red one on this plate. So what we have now, how many types of bacteria are here? Just one. So what we did was we took a mixed culture, and now we have what we would call a pure culture, which is just one type of bacteria. Just one type. The word subculture means to make a culture from your original culture. Our lab tech does that all semester long. He'll order, from the company a few cultures of E. coli, and then he'll take that master culture of E. coli and he'll subculture it. So he'll take maybe like 10 plates. He'll take his loop, he'll grab some from the one we received from the company, do a zigzag on a plate, grab another loop, do another plate, and then all the new plates he made were subcultures because they came from the original one. And then you can make a subculture of that and so on. Okay. So next up we have, so that is isolation, because you're basically separating the bacteria, and then ultimately your goal is this pure culture. Inspection. Inspection would be further analysis of your bacteria. Let's say you have that pure culture, you have just that red bacteria, but you want to know what shape it is under the microscope. You want to know if it's gram-positive or gram-negative. You want to see if it's motile or not. You want to know more about it. You can inspect it. So you can look at a number of, different types of aspects of that microbe. So that's inspection. Identification is, let's say we don't know what the bacteria is. Well, you can run a series of tests to try and figure out what that bacteria is. Number of different things that we'll do this semester. We'll do everything up here. with the exception of DNA analysis. We'll discuss it. I'll give you some examples. We're just not doing any, fingerprinting or PCR, Western blots, or we'll do ELISAs, which you're looking at an immunological type test. We'll do a bunch of biochemical tests, and then a lot with the microscope. Particularly in the beginning of the semester is relatively microscope intense. Those are your 5 I's. As far as media is concerned, so many different ways we can classify media. 3 most common ways we can do this. First one is physical state. Just what is the consistency of the media? 3 categories. You worked with 2 of these 3 categories the other day. First up, we have liquid media. So I'll write liquid media. It is just that. It's liquid. Everyone used nutrient broth. This is liquid. If I were to take the cap off and tip it upside down, I'm going to spill it all over the place because it's liquid. With this, there is 0% agar within liquid media. Agar is a solidifying agent. There is none in here, which is why it is liquid. So here we can see liquid media, no agar, no solidifying agent, okay? Water-based liquid media. So many different types you can get. Most common one we use is just your straight up nutrient broth. Semi-solid media, I'll write that on the board too, semi-solid. This one we have not worked with. This one is used for one main reason, and that is to test for motility. See if your bacteria is motile. You can bet semi-solid is somewhere in between liquid and solid. And with semi-solid, there is less than 1% agar. That's it, less than 1%. It gives it a very loose, jello-like consistency. I didn't bring this one. Oops, that's liquid. That was almost a disaster. No, that's just a deep. This is similar, this is just solidified, but if I were to shake a semi-solid, it would kind of jiggle around in there a little bit. It's definitely not liquid, but it's not as tough, as dense as your agar plate that you worked with the other day. So it is good for motility. Here we can see low percentage, less than 1% of agar, used for motility testing. So what you would do is you wouldn't use a loop. This has a loop on the end. You'd use a needle, so it comes to a point. Grab some bacteria. You stab about 3/4 of the way down, bring it back up. This is before you did an experiment. This is after. Here, you see where the bacteria grew, wherever you put it. That would mean it's not motile, because you put it here, that's where it grew. This result, you can't even see the stab mark anymore, because the bacteria are able to move out that way, move out that way. This is more cloudy than the sides here. It's hard to tell in this picture, but also you see all the growth on the surface. So this bacteria is motile, this bacteria is not. If you were to use solid, it's too dense. Even if they have flagella, they can't move away from the stab. If you use liquid, well, you just do that and then they all move, so you can't tell if it's because you shook the tube or if they're motile. So that's pointless. So this has just enough agar where you can kind of trace and see where they went. And if they don't go anywhere, it's because they're not motile. This one, depending on the type of bacteria, sometimes they give off like a sulfur byproduct, which is a dark color. So it's tough to really see much there. Last one we have, solid media. You might think there's going to be like 30% agar. No, there's really not much, but it's enough. Solid media, somewhere between 1 and 5% agar is all you need. When our lab tech makes these plates, he uses 1.5%. That's it. And this is, it's the same thing you touched the other day. This is what you used the other day, 1.5% agar. It's reversible, so if we were to heat this up, it will turn to a liquid, and we can pour it into another container. So like in here, he poured them in here, so they solidified. There's not much surface area to work with on the top of this, however. So sometimes you pour it into a tube, and then see how this one's kind of on a slant? You let it cool on a slant. That's what he did here. He poured it in, let it cool on a slant like that, and then you have all this surface area to work on. So you get all your bacteria on the surface. You never puncture it. The only time you ever puncture media is when you're doing that motility testing. The rest of the time, you would take your loop, and you just stay on the surface. That's it. Just make a nice little zigzag on the surface. That is it. Plates, again, when we did the plates on Tuesday, you weren't supposed to, push down too hard where you punctured it. You just stay right on the surface, and that's what you'll do with the loop too, right on the surface. So what I'd like to do is I'm going to pass around the slant, just nutrient agar that solidified on an angle, and the plate and the loop, just in case you want to practice how much pressure is needed to stay on the surface but not puncture it. Good practice, because when we do these labs, you never want to puncture the media. So I'll send it around if you want to try. You don't even have to. I'll send it around, at least you can see how you hold the loop and so on. Chemical composition. If we look chemically, two categories. Synthetic, all that means is no animal extract. There's a quarry across the road, and when they use the dynamite, as the building shakes. Sorry about that. So synthetic, no animal extract, or non-synthetic, that means there is an animal extract. It could be blood. We use blood agar occasionally. It could be, there's beef extract in the media going around the room right now, so it's non-synthetic. So if there's any type of animal source, non-synthetic. No animal source, synthetic, purely chemicals. That's it. Last category, functional types. Your enriched media, it just means it has, the most common nutrients like vitamins and amino acids that your standard bacteria like. The media going around the room right now, what we used on Tuesday, it's just your basic enriched media. Just about all bacteria will grow. It's not very specific. General purpose. So enriched, it's got the good stuff, amino acids, vitamins, different growth factors. There's a new definition up here. Fastidious. If you're doing some research and you see the bacterial organism is fastidious, that basically means that the bacteria is picky. It requires a certain nutrients in order to grow. It has to have something. This plate up here, plate A, this is just showing bacteria growing on blood agar. Some of the bacteria digested the red blood cells. That's why there's this clear zone around the bacteria. Kind of ate up those red blood cells, broke them down. Hemolysis took place. This right here, this is showing Neisseria growing on chocolate agar. Chocolate agar is not made out of chocolate. It just looks like chocolate because it's dark brown. All that it is blood agar that you boil. So you boil it up, it does not smell good, and then pour it into the plates, and it just has a really dark brown color. Neisseria, so Neisseria gonorrhoeae, that organism will grow on chocolate agar, but it doesn't like your standard blood agar. So it's picky in that sense. Okay, so it's fastidious. Next up, selective. Just think about the word selective. That media will be selective for some bacteria, so some can grow, others can't. If you use a media that is selective for E. coli, and let's say you have an unknown bacteria, you try to grow it on that media and it doesn't grow, you can rule out that you have E. coli. If it does grow, then you know you got E. coli. So there's a lot of selective medias out there, just depending on what you're looking for. There's a picture right here. And actually, I'll give you guys selective media on Tuesday. You'll take home something called Saburad agar. Saburad agar. It's going to be in a plate. It will only grow mold. It has different types of antibiotics infused into the agar, so bacteria will not be able to grow. So you'll take that home and you'll be able to leave the plate exposed. You leave it exposed for like a half hour or an hour. Cover it up, bring it back to class. In a week or so, you're going to have a bunch of mold on it. But no bacteria, because it's selective only for mold. So here, they're showing a mixed sample. Everything grows on general purpose. On selective media, only whatever you're selecting for grows. You choose your media accordingly. That's all that selective is. So the Saburad, the mold one, is a really good example, because we'll actually do that. We'll get to take that home next week. Last category, differential. Easiest way to remember it, everything grows, but they all look different. Everything grows, but you can see differences between the bacteria. So in this image, what they're showing, here's your mixed sample, that's your mixed culture. Here, general purpose, everything looks the same. Differential medium, everything grows, but they're different colors. That's differential. You'll have a little key that says, okay, it grows purple, it's Chromobacterium. If it grows red, it's Serratia. If it grows white, it's Escherichia, whatever it might be. So that's pretty cool, and here's an example. We'll talk about the top example. I like this example much better. This media, thank you. This media right here, this is called mannitol salt agar. It is made with like 7.5% salt. If you were to take most bacteria and plate it, put it on that plate, they're gonna die. They're not gonna be able to grow. There's too much salt. It'll completely dehydrate whatever you put on it, and you will not have growth. However, there is one particular genus of bacteria that likes it, that actually likes to grow on mannitol salt. That is Staphylococcus. So if you have an unknown bacteria, you don't know what it is, you're like, I wonder if I have Staphylococcus. Grab some mannitol salt agar, do an inoculation. If you have growth, you know you have Staphylococcus. So it's selective for staph. Great. Okay, so that tells you you have Staphylococcus, but does it tell you if you have Staphylococcus aureus? If you have Staphylococcus saprophyticus? If you have Staphylococcus warneri? Doesn't tell you what type of staph you have. This media is awesome because it's also differential. That means all staph grows, but different species of staph grow different colors. So this media is awesome because it's selective for staph, but it's also differential because different staph species grow different colors. So right away, you can be like, oh, if it grows yellow and it changes the background to this yellow-orange color, that's Staphylococcus aureus. If it grows white and the background is this pink color, that's Staphylococcus saprophyticus. If it grows yellow and it's kind of pinkish in the background, that's Staphylococcus warneri. Differential for different species, selective for staph. Awesome, awesome example. You should be able to explain to someone else, explain to me, why this media is selective and differential. What about it, is selective and differential? This right here, one of the most important slides and one of the most difficult concepts, I think, for students to kind of wrap their minds around when you're learning about microbiology. So what we have here is with this, let's do some writing on the board real quick. We're going to talk about what a colony is. So, and also what I want to talk about is what you can see with the naked eye versus what you need a microscope for. So if we are looking, let's just kind of get this picture up here. If you are looking through a microscope, you can see some of the bacteria has a circular shape. Some of the bacteria has a rod shape. Okay, you could not tell that. Like when you grabbed your slide on Tuesday from the slide box, you couldn't tell by looking at that slide what shape the bacteria had, because that's microscopic. On plates, so up here I have someone's result, this plate. I can't look at that plate and see what shape the bacteria are. I need a microscope for that. So what you need to know is microscopic are the bacterial cells. You need a microscope to see individual cells. So what happens when I have this plate and I take my loop and I do a zigzag on the plate? So let's say Laura decides to take a plate and touch her finger to the plate. Let's say when she did that, she deposited, this is her plate right here, she deposited 1, 2, 3, 4 bacterial cells. When you touched your plates on Tuesday, could you see the bacteria on your plate? No, you couldn't see the bacteria on your plate. So let's say she touched it and she deposited 4 cells, puts it in the incubator. What happens is 20 minutes later, this 1 cell turns into 2. This 1 cell turns into 2. This one turns into 2. This one turns into 2. 20 minutes later, 2 become 4. 20 minutes later, 4 become 8. After 48 hours, what you're going to see is each one of these bacterial cells turn into hundreds of thousands of bacterial cells. Once there are that many, they start to stack up and stack up and stack up, and pretty soon they turn into these big, huge mounds on the plate. These big, huge mounds on the plate we would refer to as a colony. So these colonies, these started from 1 bacterial cell. Began from 1 cell and just multiplied and multiplied and multiplied. They were in the right growth environment, turned into millions of bacterial cells. All these cells, all started from 1. Each one of these colonies started from 1 bacterial cell. In parentheses, I'll say the same type. That just means if this 1 bacteria was E. coli, the hundreds of thousands that grew, they're all E. coli. If this one happened to be staph, then they're all staph. So here we can see microbes become visible as isolated colonies. Here's colony, because they contain millions of cells stacked on top of each other. That becomes macroscopic. So the colony is macroscopic. That means you can see it with the naked eye. That is very important that you grasp that, what you can see with the naked eye, what you can't. So back here again, a colony, discrete mound of cells on the surface of agar that began from just 1 cell. Contains that species, no other. Let me do this. Liquid, solid. The liquid media used on Tuesday. When you get your results back, where is the growth of bacteria? Everywhere. It's all over. So if we look at, I grabbed one from another section. This was actually done yesterday. This is someone's phone. So if I hold this up, you can see it's even creeping up the side of the tube, all this white all around, but it's cloudy compared to that sterile one. There's a difference starting to appear. By tomorrow, there'll be even more of a difference. So basically, when you have liquid, you have growth, in the broth. It's everywhere, it's throughout. So I'll just say everywhere. Solid. Where will the bacteria grow with the solid media? On surface. So I'll say growth on surface. This is a plate from my group yesterday. This is someone's Fitbit. Already, in one day, we can see how much growth there is. Okay, so there's all kinds of bacteria going on this plate. So very, very fast, but everything is on the surface. So that's important to understand as well. Okay, I'm going to save the street plate method until when we do it in lab. Pure culture, mixed culture versus contaminated. Talked about these definitions already, just a quick review. Pure culture contains how many types of bacteria?
One.
Well, just one. This is a pure culture of whatever that white bacteria is. Maybe it's E. coli. This is a pure culture of whatever the yellow bacteria is. This is a pure culture of whatever the red bacteria is. Mixed culture contains how many?
More than one type.
More than one. So it could be 2, it could be 3, it could be 4, it could be 10. This plate is an example of a mixed culture. There's yellow bacterial colonies. And again, each one of these colonies you can see with the naked eye contains millions of bacterial cells. And there's white ones. There's white ones and there's yellow ones. These are each pure, because this is just yellow, this is just white, but this is mixed. Again, you can make pure ones from a mixed one by taking that little loop tool, grabbing just the yellow one, and dragging it across the slant. And then aseptically you flame it, kill everything on it, and then grab just the white one and put it on the slant. So you've got pure cultures from a mixed culture. Over here, we're seeing something contaminated. You're supposed to have just red, but you have this weird white blob, which is definitely contamination. If in lab you ever grab a plate and you open it up to use it and you see some mold or something on it already, throw it away in the biohazard bag and grab a new one, because it's contaminated. If you get results and you see something really odd that shouldn't be there, you would write in your lab report, it's contaminated. You still draw what you see, but you know, it would be contaminated. I mean, it just takes a second. If you leave the lid off for a second and even your hand going over it, your hair going over it, talking, that's all that it takes. So when you work with plates, just take it off very quickly, do what you gotta do, and you cover it up right away. And contamination happens very, very fast. Incubation, we talked about temperature. Okay, usual range, 20 to 40. And we talked about this too. Okay, cloudiness, if you're looking at growth in liquid, it's throughout. Looking for cloudy and type of sediment. And we saw that with this tube up here. On solid media, you're looking for colonies on the surface. Everything's on the surface. Last thing I have is just looking at size. One more time. Right here, this picture, we can see macroscopic view, or macroscopic, naked eye. This book says the range of the human eye is 1 millimeter. We know it's really 1/5 of a millimeter, 0.2, as we talked about in the beginning of class today. You could see a louse, head lice with the naked eye, reproductive structure, bread mold. Okay, you can see that with the naked eye, of course. Microscopic view, range of the light microscope. They're saying 1 micrometer. Again, we know the ones we have, we can go down to 1/5, so 0.2 micrometers. Most bacteria, again, are between 1 and 10. So definitely, standard light microscope, you can see most bacteria. Okay, you can also see things like red blood cells or algae, things like that. You would need an electron microscope. if you wanted to see viral particles. Mycoplasma, they're usually tiny bacteria, but looking at your viruses, you would need to have an electron microscope, which are very crazy expensive. Where I went to school, for example, we had one employee at the university who was responsible. Her sole job was running the electron microscope. We weren't even allowed to walk in the room where the microscope was. You just give your samples, she would run them, you get the report back. But they're pretty intense, but they are awesome. Okay, but what we can see in here with our light microscope is plenty for this class. We can see just about all bacteria, which is awesome. And then finally, check. What are the two ways we can use the term culture? What's one way? Is a noun. So this, if I had E. coli on this plate, I would say this is an E. coli culture. What's the other way I could use it? Is a verb. So I can do a culture, which means I can culture up some E. coli. That means grow some new E. coli. Do a subculture. 3 physical states of media. I have 2 of them up here. This is your liquid state. How much agar is in liquid?
0.
None. Semi-solid?
Less than 1%.
Less than 1, and then this is solid, which would have
1 to 5.
1 to 5. Yep. Selective versus differential media. Which one can you see? All bacteria, they just maybe have a different color.
Differential.
That would be differential. And then selective, just you can use it to select for one type of bacteria, one genus of bacteria, or one genus species of bacteria. So that is the difference between the two. Okay, that is everything I have for chapter number 3. Chapter 3 also goes through the details with the light microscope, but we covered that in lab number 1.